I am part of the Biomix team. Would like to welcome you all for today's talk. A personality in a platform and eminent scientist from ERA University, which is the top most leading private institution. In fact, we today have Professor Shadab Raza with us for today's talk. On behalf of the Biomix team, I would like to extend my warm welcome to Dr. Syed Shadab Raza. He is here today with us for his talk on stem cell therapy for translational neurology, for an area of stem cell therapy and regenerative biology or regenerative. So I will be showing you neurology. I'd like to thank Dr. Syed Shadab Raza. My invitation, in spite of us, it's really honored for the Biomix team to present in a virtual platform. Join with us for our presentation. You are most welcome to place your YouTube to bring a speaker, and you can place your feedbacks as comments in the same chat box of the YouTube. For the upcoming webinar talks with of the Biomix, you can in the description. Follow, subscribe for field of balance for their active participations and made this consecutive talks and subscribers in YouTube channel, and this credit goes to our broad audience, right from undergraduates, postgraduates, postdoctoral community, and all other faculties of life sciences stream who made this event a great successful. Now, let me introduce our speaker to you. Today's speaker is Dr. Syed Shadab Raza, and he is currently working as an associate professor in the Department of Stem Cell Biology and Regenerative Medicine in Iras University in Lucknow Medical College and Hospital. As an assistant dean for Faculty of Basic Medical Sciences in ERA University and coordinator of Ph.D. program in ERA University. Looks after the clues regarding the underlying fat of stem cells after transplant in an enzymic brain. His lab employs therapeutics manipulation to enhance the survival of the transplanted cells. Raza is also a visiting scientist in American University of Barbados in Latin America. He was a postdoctoral fellow in the University of Utah in USA and in Cleveland Clinic, USA. He was a postdoctoral research fellow in the University of Adelaide in Australia, and he was also an investigator in the University of Ferrara in Italy. He worked as a research scientist in Max Delberg Center for Molecular Medicine in Germany and in the University of Greifswald in Germany as well. Syed obtained his doctorate in Ph.D. in toxicology from Harvard University in India. He obtained his master's in MSc toxicology University. He has a citation of 2005 and Google Scholar had several awards, such as a Fellow of the Royal G from United Kingdom in 2020, a lifetime member of Academy of Science India, 2019, Mrs. Ariba Marke Neuroscience Award, and a Senior Research Fellowship in 2012, APRC Exchange Fellowship to Australia, recovery in 2000, University Merit Award Gold Medal, EU Marie Curie Action Fellowship, Stem Cell in Czech Republic. With the intro, now we search a speech with your telecasting in a channel, and after the completion of the video, the session will be question and answer sessions. You are most welcome to place your questions, and Syed is with us in our platform once the completely played for question and answer sessions. With this, with the permission of Syed, thank you so much.
Very good evening to all of you. First of all and foremost, I am so thankful to the Biomix and especially to Mr. Partha for giving me this opportunity to deliver a talk and share our experiences regarding stem cell therapy for neurodegenerative diseases. My today's talk would be more educative one rather than what we do in the laboratory. So quickly coming to the slide number second, this is our building. This is our campus, actually the ERA University campus. The left one is the university building, the right one is the hospital where I work, and the third one, the down one, is the college where my laboratory is situated. My laboratory is situated on the 5th floor of this building. So just to briefly introduce about our work, we are primarily a neurology lab, and our prime interest is to look into how cerebral stroke and spinal cord injury gets ameliorated through the translational interventions. For the same, we are employing state-of-art techniques like genetic manipulation, nanoparticle induced therapies, stem cell conjugated with nanoparticles to the diseased brains of rodents. Now, before I start today's talk and let you know what a stem cell is and what is the neurological disorder and how stem cell is mitigating the effect of the neurological outcomes, disorders outcomes, let me introduce you to the a couple of world renowned players who have taken stem cell therapy for their ailments. The first one on the third slide, the right one is Cristiano Ronaldo. You are well aware of him. He was the Portugal national team captain, and he has a broken ligament, and he took stem cell therapy for that. Similar was the case with the Ronaldo Cristiano, who has a broken muscle in the leg, and he has received stem cells. And Ronaldo has revealed that prior to taking stem cells, he has never experienced such a benefit at least 2 years prior to taking the stem cells. And then in coming to the next slide, this fellow on the left of you is Bartolo Colon, who is a baseball player of United States, and he had also a broken ligament and took stem cell therapy for the broken ligament. And the last one is Sam Harrell. He is the father of Graham Harrell, renowned football player. Sam Harrell has to quit the football coach job because of the debilitating symptom of multiple sclerosis. And while taking this treatment, after a couple of years of taking the treatment of stem cells, once he said that I am the luckiest guy of this world, as stem cell has given me my life back. Now, a brief introduction of the history of stem cell therapy. It was 1968 that the first BMT therapy, that is the bone marrow transplantation therapy, was used for a SID, that is a severe combined immunodeficiency syndrome in an infant. It was not earlier than 1998 that the embryonic stem cell were identified. And very recently, that is in year 2006, it was Shinaya Yaganama who produced iPSCs, that is induced pluripotent stem cells from the somatic cells. Now, coming to the next slide, the reality versus the hype of stem cell therapy at clinics. So, I am sure that you must have gone through several advertisements from India and as well as from across the globe, that several clinic claims that they can cure multiple ailments or multiple disorders with the use of stem cell therapy. Unfortunately, this is far from the truth. Honestly speaking, in our view, the stem cell treatment should be viewed as more experimental rather than to be used in standard practices. Stem cells have tremendous potential to understand and treat a range of diseases. Before moving to the next slide, I would suggest that to be very cautious before you go to a private clinic which make claims for treating a variety of diseases employing stem cells. Now coming to the stem cell approved therapies, to date the FDA, that is the Food and to date the FDA, that is the Food and Drug Administration of United States, has just permitted homeopathic or bone marrow stem cell therapy, and that too only to be used in cancer ailments. So what does it mean? It means that the use of any other sort of stem cells or their subtypes in any other disease other than the cancerous stem cell therapy is not approved by FDA. Well, noteworthy is that California's stem cell agency has a list of disease where you can employ stem cells, and the list of the stem cells or the other cellular therapy that can be employed into the clinic. So similar to the FDA in United States, which regulates the stem cell therapeutic use in the clinic, we have NAC SCRT, that is National Apex Committee for Stem Cell Research and Therapy. So that governs the preliminary regulation of stem cells to be used in clinic as well as to be used for basic research purposes. So coming to the current status of stem cell therapy in India, as far as I am aware of, there is no permission of employing stem cells in neurological diseases at the clinics, and similar to the FDA, the NAC SCRT also permits the use of stem cell for hematological diseases. Here I would like to mention that mostly in clinics the SCT, that is the stem cell transplantation, is equivalent to bone marrow transplantation, since it is the only one cell type that is permissible to be used. So if you will ask me to give an example for the same, so I will say that suppose if a person's bone marrow is not efficiently making blood cells, it will eventually lead to anemia and profound bleeding. So in that case, we can employ hepatic stem cell to restore the blood population of that particular person. Now next slide. Well, irrespective of the source from where the stem cell has been derived, so there are multiple organs from adult itself. You can have stem cells from bone marrow, you can have stem cells from hairs, you can have now with the invention of this iPSC technology, you can have stem cells from your somatic cells also, for example, like your skin. So irrespective of from where we have derived the stem cells, a cell to be recognized as a stem cell, it should have at least two minimal and essential properties. The first one is that it should be it should have a property of self renewal. So what does it means? That that a cell, when it divides, it should produce exactly the replica of the same cells. So that is the one property that is called self renewal. The another one that is that it should have the property of showing multipotency or pluripotency. So multipotency is a property of the cell where a cell can make multiple type of cells but not all type of cells. I mean many type of cells but not all type of cells. Unlike pluripotency can give rise to any type of the cells. So this should be you must have in your mind that a cell to be a stem cell should have at least two properties. The first one is that it should be should have a property of cell renewal, and the another one is the property of showing multipotency or pluripotency. Okay, if you will ask what could be the example of a multipotent cell and what could be the example of a pluripotent cell, then I will say, for example, the bone marrow derived stem cells. So bone marrow derived stem cells has is a adult stem cells. We will shortly deal with embryonic, adult as well as iPSCs. So bone marrow derived stem cells is a adult stem cells. So it can produce any type of blood cells, and the another good example is neuronal stem cells. So a neuronal stem cells is another type of adult stem cells, and it comes into the adult category, and it can produce neuronal cell as well as oligodendrocytes as well as glial cells. So, this is called multipotency, that neuronal cells can give a variety of cells, but not the all cells, for example like the embryonic stem cells. So, embryonic stem cells can give rise to any part of the body. It can give rise to the cells of cardiomyocytes, it can give cells of liver, it can pancreas, brain, etc. Since the whole tissue of the human being is made up of embryonic stem cells. Now, coming to the next slide, this slide is very important for if a cell has to be used in the clinic. So, what are the prerequisites of a cell for clinical transplantation? The very first thing is again the self renewal. It should have a property of clonal propagation. So, it should be able to produce the exact replica of the cell. The another very important thing is that it should be genetically stable. The another important aspects are that they should be capable of integrating into the host tissue. For example, if I transplant a neuronal stem cell or mesenchymal stem cell to the brain, it should go and get integrated into the brain, either in the form of neuronal cell, glial cells, or oligodendrocyte, whatever, etc. The another thing is that it should show the capacity of migration. So, once we transplant those cells in, they should be able to reach to their destination. The third very important aspect is that they should be able to differentiate into the particular cell type of that particular organ. For example, I said the neuronal stem cell should be able to be differentiate into astrocytes, glial cells, oligodendrocytes, neurons, etc. And finally, at the end you will ask what is the outcome. So, in terms of functional benefits, so it should be functionally beneficial with having minimum side effects. Coming to the slide number 13. So, this is a beautiful presentation of how stem cells can produce a variety of cells. So, first of all, let us see the number 1 figure, which shows embryo. So, what happens when a sperm meets a ova, it produces zygote, and zygote eventually give rise to embryo. So, embryo at the day 1 to 5 produces a lot of, they get divide and produces a lot of cells, and at the day 5 to 7 they form a spherical structure which is called blastocysts. So, blastocyst, the inner layer of the blastocyst contains a layer of inner mass of the cells, which are basically embryonic stem cells. And these embryonic stem cells, when taken into the petri dish, of course, you have to destroy the blastocyst at this stage, and when you will put it into the petri dish, you can at laboratory you can grow embryonic stem cells to a variety of tissues. For example, they can produce the cells of pancreas, they can produce the cells of kidney, the heart, neuron, brain, etc. Now, the very next slide shows the stem cell types depending on the source from where they are taking. So, the third figure shows a embryo. So, these are called embryonic stem cell. The cells derived from this embryo is embryonic stem cell, just I showed previously. The embryonic stem cells are largely of pluripotent nature. We can also have pluripotent stem cells from zygote, but the cells from the zygote has a limited potential. They can form placenta, they can form zygote, of course. And the fifth figure shows the embryonic germ stem cell line that is derived from the aborted fetus. And now, coming to the fetus gives us pluripotent as well as multipotent cell types. Also, you can have stem cells from adults, which are known as adult stem cells. So, broadly they are divided into two major categories, the embryonic stem cells and the adult stem cell. And the third one, since 2006, since the discovery of the iPSCs, the third one is naturally IPSC, that is induced pluripotent stem cell, that those can be derived from somatic tissue with the help of some of the reprogramming factors that I am going to talk right away. But before that, please look into the next slide, where we have a beautiful differentiation between the embryonic stem cells as well as adult stem cells. So, embryonic stem cells, as the name signifies, it is derived from embryo, and the adult stem cells comes from adult tissues. So, the nature of the embryonic stem cells are largely pluripotent, and the nature of adult is multipotent. So, they can give multiple cell types. However, the adult stem cells are less stable compared to the embryonic stem cell. However, the adult stem cells are less stable compared to the embryonic stem cell. What does it means? That embryonic stem cells can give rise to multiple cells. They can divide and divide, which is not the case in the adult stem cells. But the best thing with the adult stem cell is that they have no ethical issues, which is often associated with the embryonic stem cells. However, the adult stem cells are difficult to isolate compared to the embryonic stem cells. Now, let us talk about the induced pluripotent stem cell in brief. So, as the name signifies, it is a form of induced pluripotent cells, and the best thing of these cells are that they can produce all the three germ layers, that is ectoderm, mesoderm and endoderms. Now, the next slide shows how Yakanama produced iPSCs from mouse fibroblast cells. If I will go into the detail, initially they have taken approximately 22 factors, that is the reprogramming factors, and then they introduced those factors to the mouse fibroblast cells, and eventually they came up with the four major reprogramming factors that were responsible for in changing the somatic stem cells to the iPSCs, and those were KLF4, SOX2, OCT3x4 and CMYC. So, these four set of reprogramming factor when introduced with the mouse embryonic cell line, fibroblast cell line, they eventually give rise to a variety of adult cells. So, the present slide depicts how iPSC can eventually give rise to cardiac cells, brain cells, pancreatic cells and adipocyte cells, etc. Now, coming to the next slide, this shows a iceberg, and what it shows actually that we are dealing with a system that is extremely complex, the CNS, that is the central nervous system. If you will ask me about the neuronal population of the CNS only, then it accounts for approximately 85 billion to 100 billion. So, you can just imagine the Milky Way, which has been vastly studied, has approximately 200 to 400 billions of stars. So, you can just imagine the complex architect of the brain and the spinal cord. Now, coming to the neurological diseases could be either due to the injury or due to the progression of the neuronal degeneration, or it may be due to the disorder of development, for example, the Down syndrome. Now, a very important question is that why we need stem cell transplantation therapy for the brain. So, it all started as back as in 1913 when Spanish scientist Santiago Ramon Cajal pronounced that in the adult neuron centers, the nerve paths are something fixed, ended and immutable. So, he believed that everything may die and nothing can be regenerated. And this perception extends for at least since 1913 to 1965, when Das and Altman in 1965 first shows that there are the sign of regeneration in brain too. So in 1965 Altman and Das first identified the subventricular zone, a zone which was capable of producing new cells, that was neuronal stem cells in the rat brain. Therefore, the perception that once a cell of brain is died, it is died forever and it could not be generated again came to a halt, and it was identified that there is at least a couple of zone, subventricular zone followed by the dentes gyrus of the hippocampus. So these, at least in these two regions, the cells can, the neuronal stem cells can be produced by the diseased or the healthy brain. Now, the next very important question is that why the stem cell therapy is getting so much of attention in the neurological disorders. Now, the next very important question is that why stem cell therapy is getting such much of attention for the neurological disorders. So, as I said, the very first thing is that it is same for all sort of therapy. So, it is due to their capacity to proliferate, their ability to migrate and to integrate into the system. The another important aspect is that they may show tropism in the diseased brain. Further, they have the ability to show transdifferentiation. So, if you will ask what transdifferentiation is a property of an adult stem cells. So, when they are isolated from a particular region and transplanted into another particular region, they can generate the cells of that particular organ. For example, if you are taking out bone marrow derived stem cells and you are injecting it to my brain, they have the ability to produce the cells of brain, that is the neuron, glia, etc. So, this is called transdifferentiation or plasticity of the stem cell. Specifically, this is associated with the adult stem cells. The another very important possibility that is associated with the stem cells are they can easily be manipulated via the use of non-viral as well as viral gene transfer methods. Now, the next slide is from our lab, and it shows that how a stem cell, in this particular example we have employed dental pulp stem cell, that but this holds true for most of the cells. So, how exactly an stem cells give benefits to the diseased brain or CNS. So, once transplanted, either a stem cell can produce new cells, and this process is called neurogenesis, or they can form new arteries, that is called arteriogenesis, or they can help in synapse formation with the process is called synaptogenesis. And a very important aspect is that they can show immunomodulation through decreasing the pro-inflammatory cytokines or proteins while enhancing the effect of the anti-inflammatory cytokines. Likewise, they can reduce the rate of apoptosis in the cells, they can enhance autophagy or vice versa depending on what is required. Now, in the next slide, let us see the challenges associated with the stem cell therapy. So, in our experiences, what we know is that there are two major hurdles in stem cell transplantation therapy. The first one is that once you inject the cell, they have a minimal ability to reach their target organ. So, they show a very less target migration. So, if you will ask that how much of the cells which were transplanted in reaches to your brain, I will say that approximately 90% of the cells get excreted out through kidney and the lung, So, 90% is a huge loss. So, we are left only with the 10% that really reaches the brain. But for example, if the brain is your diseased organ or injured organ. Now, the problem is that whatever reaches to your brain or any diseased organ, due to the toxic and the inflammatory environment in that particular organ, it has been shown, especially I'm quoting a report of Lee and Kopp published in Lancet for the stroke repair, that out of that 10%, only 1 to 3% of the cells survived after 28 days, that is 4 weeks of the transplantation. So you can just see that how much minimum the rate of survival is there, and the retention property is there. So now you may ask me that why there is suboptimal effect of the stem cell transplantation into the organism. So the issues involved with the cell therapy, or so I will say that there are a number of unresolved issues till date, as we do not know that which is the best cell to be transplanted in, what is the best route to be chosen, what is the what should be the frequency of the transplantation, and what is the safety parameters that we have to take into the account in terms of the outcome. So these are the few things that has to be taken into account prior to the infusion of a stem cell for a particular patient at the clinics. Especially if you will talk about the clinics, I would like to mention you that at clinics there have been numbers of clinical trials going on, and there have been a lot of clinical trials already been done, but what we know from these trials, that all of these trials have indicated about the safety of these cells rather than the efficiency of these cells post transplantation, that how they irrespective of the disease I am talking about. Now coming to the next slide, as I said, the neurological disorder, which is the disease of the central and peripheral nervous systems, are because of either of injury, or because of the neuronal degeneration, or because of the tumor formation, etc. In these slides we will deal the neuronal degeneration due to the injury, and the neuronal degeneration due to genetic background. So let me start with stroke. So coming to the stroke, stroke is due to the low supply of oxygen and nutrient to a particular area of the hemisphere of your brain. So that eventually leads to paralysis or plegia in the patient. Broadly speaking, the stroke are of two types. A majority of a stroke are of ischemic type, which accounts for approximately 85 to 87%, and rest being the hemorrhagic stroke. So the ischemic stroke is due to the lack of blood supply due to the stiffening of the artery, while the hemorrhagic stroke is due to the bleeding of the condition arose due to the bleeding of the rupturing of the artery that leads to the bleeding of the, while hemorrhagic stroke is due to the rupturing of the artery that leads to bleeding in the brain. However, a common thing in whether it is a ischemic stroke or whether it is a hemorrhagic stroke, that they'll, in both the conditions, cell lacks blood and nutrients. This particular figure on the left shows blockage in the artery, while the right shows the hemorrhage in the brain. Now coming to the current treatment of ischemic stroke, if this is a ischemic stroke, we have unfortunately we have only one drug that is FDA approved, and that is known as RTPA, that is recombinant tissue plasminogen activator. The FDA has given approval for RTPA in 1996, and since almost 2.5 decades is passed, and we do not, unfortunately we do not have any other drug for ischemic stroke. However, aspirin and other blood thinners are used for in the patient of stroke which has to persons who are susceptible to stroke. However, removing of the blockage by surgery is another mean to get rid of the outcome of the ischemic stroke. Similar is the case with the hemorrhagic stroke, where surgical intervention is a must. Now coming to stem cell therapy for ischemic stroke, if you will go to the PubMed, you will find numerous papers employing stem cells for stroke recovery. And a variety of cells have been used. For example, bone marrow derived mesenchymal stem cells have been extensively used, neuronal stem cells, dental pulp stem cells are the other cell types that have been used for stroke recovery in the experimental models of stroke. And a lot of groups have shown different mechanistic approach to rectify or to derail the effect of ischemic stroke pathophysiology in the brain. Notably, the paracrine effect have been shown by various group, the transdifferentiation effect have been shown by various group, and also the mitochondrial transfer apart from the integration of the cells have been shown. So here I would like to let you know what exactly a paracrine effect is. Paracrine effect is also known as bystander effect. So in this, rather than cell goes and get integrated into the system, it produces or it secretes some cytokines, some growth factors, some proteins that can mediate neuroprotection by integrating into the system. Similarly, mitochondrial transfer, as you are well aware that in a stroke there is a loss of respiration. So for respiration, mitochondria is involved. So a lot of groups have shown that once the stem cells are transplanted in, they rather than differentiating into or integrating into the system, they release their mitochondria to the other cells so they can acquire it, and they can produce more respiratory substrates. Now coming to the next slide, that is for traumatic brain injury. Traumatic brain injury is a sudden damage due to a jolt or a blow to the brain. The common cause include accident, a fall, or play injuries, etc. Well, it is very important to mention here that brain trauma could be of because of the two reasons. Either it is due to the close head injury where something is stuck to your head but your cranium is not open, or either it could be due to the open head injury. So a very good example of closed brain injury is your football ground. If you are aware of a couple of years back, European Union has came with a resolution where they want to ban the head kick, because it was noticed at that time that a lot of players who were involved in this football practices and were giving a head kick have shown symptoms of memory deficit, mood swing and frustrations. Another very important example is your soldiers who often meet combats. So during the combat operations, it might be possible that they do not get hurt through a grenade or through a bullet directly, but in turn what happens that when for example a grenade explode in their near vicinity, so what happened that they produces shock wave, and that shock wave is enough for army personnel to get brain trauma, which could be either is of mild type or severe type. Normally mild type of the traumatic brain injury is defined when the unconsciousness or the confusion persists for less than 30 minutes. But when this is more than 30 minutes and the memory deficit is more than for 24 hours, that comes into the severe category of the TBI. So what is the option for a TBI patient? Unfortunately, there is no treatment available except the rehabilitation. Rehabilitation is the only way which can give some symptomatic relief to the patient, and later on some surgical intervention to reduce the severity of the damage could also be employed in this case. Before I move to stem cell as a therapeutic option for TBI, it is very important to mention that traumatic brain injury is the third largest cause of death due to any sort of injury. Now coming to stem cells as therapeutic for traumatic brain injury. It is again the same when you transplant and stem cells, either they can integrate into the system, they can produce new new gilias, or they can secrete some growth factors that eventually helps in restoration of the neuronal architect. Or the another way is to enhance the population of the endogenous stem cells. That could be done through giving factors which are responsible for enhancing the production of the stem cells. For example, BDNF, granulocyte colony stimulating factors. BDNF is brain derived nerve growth factors, etc. Now the next slide talks about the Parkinson disease. I am sure that you must be well aware of the Parkinson disease. You must have heard about Parkinson disease. It is a disease characterized by tremor in the body, with stiffness, problem in talking, walking, and in coordination. So, what is the cause, pathophysiological cause of Parkinson disease? So, at the molecular level, if you will see what happens in Parkinson disease, that the Parkinson disease progressively affects basal ganglia and substantia nigra. So, substantia nigra is the center for the generation of dopaminergic neurons, which produces dopamine. Dopamine is responsible for your attention and coordination of person. So, dopamine is a neurotransmitter, as you are well aware of it. So, in the patient of Parkinson disease, the alpha-synuclein monomers changes into the oligomers, which eventually give rise to a fibril-like structure, which is known as Levy body. And the Levy body is the hallmark of Parkinson disease. At the molecular level, we check it through Levy bodies that a patient or a rodent is suffering from Parkinsonism. So, what are the current treatments available for the Parkinson disease? Either you get levodopa, which is a dopamine enhancing drug, or dopamine, or monooxidase B inhibitors. However, these drugs are only be used as a symptomatic measure to reduce the symptoms of Parkinson's, such as tremor and coordination, etc. Again, here a variety of stem cell studies have been done at the naturally at labs, and the outcome of these studies have shown that either you can directly transplant stem cell, a variety of stem cells are available for Parkinson disease too, or you can in lab you can differentiate your stem cells into the dopaminergic neuron or the neurons of the substantia nigra, and you can transplant it back into the rodent, which is of course not possible in patient up to this moment. So, stem cell transplantation is the only feasible method as far as the cell therapy is concerned for a patient at the clinic. Now, the last disease that I chose for today's talk is Alzheimer disease, which is a common cause of dementia and is a progressive disease where the neurons get destroyed and eventually they die. So, at the pathophysiological level, if you will see, the signature lesion of Alzheimer disease is neuritic plaque formation and neurofibrous tangle formation. So, if you will do the histopathological analysis of the of a patient with Alzheimer disease versus the normal person, you will find there is a shrinkage of the hippocampal area. The cortex area is shrivel, which is responsible for thinking. Similarly, the histopathological analysis of the ventricle region will show the accumulation of cerebrospinal fluid there. So, again in this case the treatment is symptomatic. We have two commonly used drugs. The first one is cholinesterase inhibitor and the another one is the memantine. Like the previous cases, in this case also a variety of stem cells are available for the clinical transplantation. However, yet no clinical transplantation has been done except the clinical trials which are approved by NIH. And the experimental studies have shown that once the stem cells are being transplanted into the Alzheimer brain, they have secreted variety of growth factors. Apart from this, they were able to integrate into the system, and a better synaptic connectivity was there between the cells. Nevertheless, like in other diseases, here also several papers are there which will let you know that employing the colony stimulating factors, the nerve growth factors, you can induce endogenous stem cell growth. Here I will stop with the educative slides that I have shown. Now I wish to show some of the results from my lab and what we are doing. Already I believe I let you know that we are basically a neurology lab, and we are primarily interested in stroke recovery. So a lot of the stroke work is going in my laboratory, and the intention of my laboratory is to look into what is the fate of a stem cell post transplantation in an ischemic brain. So as I said earlier, there are two main hurdles that a stem cell has to face before it give benefit to the patient. The first one is that it lacks a target migration, and the second one is that their survival rate is very low. Fortunately, our lab is working on both of this aspect. So let me show you a glimpse of some of the work that we are carrying out in our laboratory. So the very first question that we have in our mind when we started our lab, that what is the effect of a oxidative and inflammatory microenvironment, that such an environment that exists in an ischemic brain. So what is the effect? So to mimic the effect of ischemic stroke brain, we are employing oxygen glucose deprivation model, which is a well characterized model to mimic ischemic stroke in vitro. So here what we do, we have a oxygen chamber where we make cells deprive of oxygen while the CO2 and nitrogen has been provided as it is in air. Also, we deprive our cells with glucose, since in stroke there is a deficient of oxygen as well as nutrients. So the brain cells are unable to take glucose. So we deprive cells in vitro with oxygen as well as glucose. So now I will go very quickly. The first slide shows the effect of OGD, that is oxygen glucose deprivation, on the cells. So what we have found that when we inserted the cell OGD, they have shown a different morphology than the normal one. Also, there's the cell number was reduced. The sign of toxicity was quite evident in the cells, and the best thing was that as long as we were giving the exposure, the cells were showing more toxic morphology. As you can see in this slide, we have done AE versus AO staining, which is widely used for cell death. The cell death number was increased when the time of exposure of OGD was increased. So a very important thing that I would like to tell you is that a majority of stroke that happens in human patient is of permanent type. So there is a lack of oxygen forever, you know. So that is our, that's why we had an intention to enhance the timing of the oxygen glucose deprivation. Now, implying flow cytometry, we wanted to look whether ROS is playing a role, because ischemia or perfusion often leads to oxidative stress, and in oxidative stress ROS has to play a major role. So for that we have employed flow cytometry, and our results indicated that indeed ROS is playing a major role in the impairment that has been observed in the cells. So we have used the activator as well as the inhibitor of superoxide as well as H2O2 to see the effect of the participation of the free radicals in the cells. And coming to the next slide quickly, we have further, we were interested in looking into whether oxidative stress has anything to do with the cells in an infarct brain, and therefore a battery of test for different proteins have been employed which are associated with the proteins which are associated with oxidative stress. For example, we employed ORP, that is oxygen regulatory protein, NOx2 which is a superoxide and H2O2 generator, SOD which quenches superoxide, and catalase which is inhibitor for H2O2. Also we have employed HIF as a primary marker for hypoxia in the cells. So our result cumulatively have indicated that indeed oxidative stress is playing a role when there is a in an ischemic microenvironment. Oxidative stress is playing a huge role in the impairment and the cell death number that we have observed in our studies. This was just a basic of one of the study that we are doing for evaluating the cell survival. We have also come up with a new model to study the movement of a stem cell. I will show you that one. So this particular slide shows that we have employed 3-day chick embryo to study the migration of stem cell in the chick. So the first figure, if you will see, is the 0-day egg that we are getting from hatchery, and after wiping it with 70% of alcohol, we are putting it into 65% humidity and 37-degree incubator, and we leave it for 1 day, and then after that we took out some of the albumin to create a since the egg is in oval shape, so when the embryo will start getting forming, it will often stuck to the wall of the eggshell. So so what we do normally, we took out 5 to 6 ml of the albumin. This is called layering, so that a space is being created between the wall and the albumin bed. And then after 3 days, when the circulatory system is somewhat well developed, the right vitelline artery and the left vitelline arteries are well developed. Vitelline arteries are the arteries which are responsible for bringing oxygen and nutrition to the embryo. So at the day 3rd, we open that egg, and this is called windowing, and at the day 3rd, we induces ischemia and reperfusion in the egg, and eventually later we proceed downstream pathway analysis, and with the aim to inject stem cell into the left side of artery. Remember that we induces ischemia in the right artery, and our aim and we are transplanting stem cells in the left artery. So with the aim that they should whether they have the ability to move to the ischemic region or not, to see this, we have chosen left vitelline artery for stem cell transplantation. Let me show you this video exactly how we have done it. Now coming to the next slide, as initially I said, we are employing nanoparticles, we are employing genetic manipulation. So this slide shows the lentivirus mediated genetic manipulation in the stem cells, so that we can trace out where exactly the cells have gone, and we are also introducing some transgene into the cells for our further studies. This was all for today. I hope you would have enjoyed the session, and if you have any question, please do let me know. My email ID is provided in the first slide of the today's presentation. So please do write to me if you are in Lucknow, or if you are from Lucknow, you can always you can come to my lab and visit us, and can see what we are doing apart from these studies. For your further reading, I have given some references in this slide, so please follow the link if you are interested in Indian stem cell ethical regulation. Please go to NAC SCRT link, etc. And now coming to the last slide, this is my existing team. Santosh is a postdoc for the last 2 years in my lab. Ravi is a Ph.D. student for 2.5 years. For 3.5 years, Naim is working as a PhD student, and others are research assistant in my lab. With this word, I would like to say again thanks to all of you for your quiet and patiently listening. I hope you have listened it patiently, and also again my sincere thank to Partha for giving me this opportunity. I hope we will meet again. Please do write to me if you have any question. So with this, thank you very much. So, will I say something? Should I say?
Okay, so yeah, sure.
So, okay, thank you very much, Partha, for giving me this opportunity, and to the whole team of yours. And I'm also thankful to the audience, whoever have came today to listen to this talk. My apologies that initially I thought that I would not be able to come on time, and that's why we have delivered this talk in a in an our animation department. That is from our animation department. We have a very beautiful animation department, and that I supplied to you initially. So what was your first question, please?
Yeah, let me forward the first question. The first question is that what is the benefits of stem cell therapy for autism?
Autism. So basically, if you will look into the into the molecular pathophysiology of autism, it is due to the calcium disruption, or calcium influx, I believe, and that basically leads to dysregulation. So if you will see one of our own slide that we have actually my laboratory is interested in looking into the various aspect of various mechanism that is associated with stem cell, that how stem cells are giving benefit. So we are working on neurogenesis, arteriogenesis, synaptogenesis. So one video that this presentation was on how stem cell is giving benefit in the CNS recovery. There we showed that either it could be through neurogenesis, through arteriogenesis, or synaptogenesis, or immunomodulation. So since the autism is associated with synapse irregularities, so stem cell can help in better synapse connectivity. So I mean that this is well proven. Even in from our own lab, that a paper being published in Stem Cell and Research Therapy in 2000, where there we have shown that how stem cell is helping in synaptogenetic formations. So that is the one way. And if eventually if a cell has gone, the stem cell has the potential to go and integrate into the system. So that is another way. But the initial thing is that through the pyrakine effect, stem cell helps in synaptogenesis genesis. I believe that I have explained parakine effect. Parakine, or the bystander mechanism, is that initially it was thought actually that once a cell is transplanted, a stem cell is transplanted, it goes and it integrates into the system. But later with several reports came out that it is not only the way that neurogenesis or the stem cell integration is not only the way, but basically they secrete some factors, and then eventually they go to the site of to the site of injury, and then they make synapse formation, and they make they help in arteriogenesis and immunomodulation. So this process is called parakine effect, when cells secrete some factors and they go and repair the system.
Yes, sir.
Yeah.
I'll pass on to the next question. The next question is that what are the chances of stem cells becoming cancerous or not?
Yes. So, like stem cells becoming cancerous is very, I mean, I will not say very common, but it is a common phenomenon when you are employing embryonic stem cell. However, in if you will, like in rodents, you can use, in a laboratory, you can use embryonic stem cell, but in the patients, it is not permissible to use embryonic stem cell. This is because this is the one major reason, being that they may turn into the tumorous cells, so that is why we do not use the embryonic stem cells. I think this was your question.
Yeah, yes.
Yeah.
Shall pass on to the next question. Can we use liver
Yes.
stem cells for cure any disorders? Is it possible?
For
For any disorder, can we use liver stem cells?
exactly, you can use initially I said that there are two categories of stem cell depending upon the source from where you are taking it. Either it is embryonic stem cell or it is adult stem cell. The property of adult stem cell, I mean the best thing of this adult stem cell, that they show transdifferentiation or coplasticity. So once you will isolate a cell from the heart, and you transplant into the brain, or it is vice versa, or you will take it from the liver and give that particular adult stem cell the environment that is if suppose if liver stem cell has to be transplanted into the brain, and if you will give artificial environment, even in the culture of brain cells, so it will turn up into the brain cells. So this is the beauty of the adult stem cells. From wherever they are coming, that doesn't matter, but from where you are going to transplant it, I mean they eventually take up the environment and start producing those cells of that particular region. Hello.
Yeah, 1 minute, sir. 1 minute.
Yeah.
Let me pass on to the next question. Can totipotent cells can be altered with use of stem cell therapy?
You mean the zygote?
I believe
sir.
they mean zygote. Yes, it
Yes,
has
sir.
been. Yes, indeed, yes. There, I do not recollect exactly the report, how many reports are there, but there are reports that they have made zygote cells from the embryonic stem cells.
Yeah. That will there any chance that these pluripotent stem cells be able to cure chronic kidney conditions to get over dialysis?
Sorry, I did not go. Yeah, sorry.
Yeah. Thus, these pluripotent stem cells be able to cure chronic kidney conditions, dialysis.
Pluripotent stem cell. Hopefully, yes. I believe yes. Man, a beautiful example is that iPSCs. So you can grow them induced pluripotent stem cells. So they are the main source of pluripotent stem cells. So unlike added stem cells, which are multipotent. Either you can use embryonic stem cells or you can use iPSCs, the induced pluripotent cells. So I mean, embryonic stem cell is ethically not permissible. However, with the permission of with the clearance of ethical committees, IPSCs could be used, and I believe yes. So I'm not like I haven't gone through any report of that particular area, but I think yes.
Yeah. Shall pass on to the next question.
Yes.
Next question is that what are the likely side effects of giving dopaminergic anagonistics because they can induce drastic changes in neurophysiology.
Again, sorry. I'm so sorry. What are the?
Yeah. What are the likely? Let me turn on the captions. If you options called turn on caption, it would be better to turn on caption from your side.
Caption. You mean in the audio, in the video?
Oh, yes, yes. Do you find any options nearby video? There is a video option, right?
Yes.
Nearby video option, there will be a turn
Turn
on
on.
caption.
Yes.
So that it will be there whenever whatever speak, you can see as a text over there.
Ah, great. Thank you.
Yeah. Now we do now problem resolved, I think. So let me put the question again. 1 minute. Yeah. The question is that what are the likely side effects of giving dopaminergic anagonistics because they can induce drastic changes in neurophysiology?
To be honest, I'm not sure, and I'm not the right person to let you know what are the, because we do not work with the with the drugs, in fact, or their inhibitors, in fact, and also we do not have, like, we do not work with the Parkinson disease. Which will do with the Parkinsonism, I am not the right person, to be honest.
Okay, so no problem. We shall pass on to the next question.
Sorry.
Shall pass on to the next question, sir. The next question is that can we grow stem cells by the expression of its gene?
Which gene? Which gene?
Of its gene, its own gene.
Which?
By identifying a particular gene which is responsible for synthesis of a particular stem cells, the question is based on this. Is it possible to grow a stem cell by the knowing the expression of that particular gene?
Yes, there are, especially if you, if I will say, for example, if you want to grow a stem cell from your blood cell, then I mean, for whatever cells, Yakanama, who has got a Nobel Prize in 2013, was due to the discovery of these four. There were four factors, basically, only four genes initially. That they found that they would be able to produce stem cells in laboratory. So those four genes I have mentioned somewhere. Let me tell you. The one was KLF4. The another was SOX2, and were there. So that I have shown in the lab, in my presentation itself. Yes. So the another one is CMYC, and the fourth one was OCT3x4. So these were the four major genes that are when uh, subject cells with these four reprogramming factors, then it will eventually produce some iPSC genes cells. Yes, and then these iPSC cells will turn into the the normal cells will turn into the induced pluripotent stem cells. And one of the slide actually.
Yeah, this or these programming factors are very specific for blood cells.
That cells means which cells? No, yeah. Let me tell you. Okay, I got it. So this reprogramming factor will change a normal cell, a somatic cell, into a stem cells. That is, and that those stem cells will be called as induced pluripotent stem cells. So that is an induced pluripotent cell is actually equal to the embryonic stem cells. Now, since the embryonic stem cell has the property to make any cells of the body, so like embryonic stem cell, since the iPSC shares common common properties like embryonic stem cell, so they can eventually give up any type of cell, whatever cell you want. Finally, the somatic cell, whatever cell you want, no neuronal stem cell you want, cardiac myocytes, whatever you want, you can eventually make those cells from these iPSC cells. So the first initial thing is that from when you programming factors to a normal somatic cell, for example blood cell, for example your skin cell, whatever, your hair cells, whatever. So it will eventually change those cells to the induced pluripotent stem cells, and then from induced pluripotent cells, whatever you want, you can giving the providing media, you can make whatever cells you want to make.
Yeah. Thank you, sir. I'll pass on to the next question. The next question is that regarding stem cell therapy, how are microglial efforts be controlled in clearing of the external cells injected?
Microglial cells.
Microglial efforts be controlled in clearing of the external cells. Whenever we inject external cells, how these microglial effect in controlling the
I
therapeutics.
Microglial cells, how they are what happens, for example, if I will give an example of the brain. So what happened that when there is an injury, the microglial cells or the macrophages, they try to engulf those because the stem cell, when you will transplant a cell, if it is not allologous, it is autologous, that is, it is coming from a different species, or a different like from if I am getting cell from your cells, so microglial or macrophages will try to to engulf those cells, as they will identify those as external cells. Okay, so that will eventually to if you want to those cells to be there and to integrate into the system, for that you have to give some immunosuppressant, so that these microglial cells or the macrophages will not be able to identify your cells.
Yeah. The next question is that will stem cell therapy be more useful as compared to CRISPR technology in case of nerve tumor?
That to either deplete that particular whatever the nerve whatever the gene is responsible for tumor, or you said about the nerve tumor, no? Nerve tumor.
Yes, sir. Yes.
So, in my opinion, the stem cell therapy is not given for a in tumor cases. So I mean, CRISPR-Cas9 is always a good choice for this sort of therapies for tumor cells and tumor cancerous cells. So in my opinion, we never give stem cells for a tumor.
Yeah. Okay, sir. Let's wind up with this with the lecture-based question. Shall enter into some experience of questions from on based on your experiences. Shall we?
Sure, yes.
Yeah. Very recently in 2020, sir, you got a member, a fellow of the Royal Society of Biology from
Yes.
United Kingdoms.
Hmm?
How do you feel and for this recognition, and what do you think, what ad made you to get into this?
Well, naturally, we are happy with. I'm happy with this fellowship. This is basically the fellowship, the fellow membership that I received from Royal Society is a recognition that you are well aware of your subject, and you are that you have a good understanding of your work and your subject. So I believe that. Our work that we have up for this cheek embryo and the stem cell therapy, because the problem is that the problem with the stem cell therapy at clinic is that there is a very low retention of the cells, and there is very low survival of the cells. So my laboratory, we are interested in looking into why there is a low retention and why there is a low survival. To take up these two questions, we have from the day zero we are working on on the homing of the stem cell, that what makes a cell to home more to their target organ, and the another is that how we can enhance the survival of the cells. So I believe that might have attracted those people to accord me this fellowship this time.
Yeah. Thanks so much, and our congratulations on this to you.
Thank you.
Yeah. Shall pause for the next question. Over this pandemic, I come to understand your citations rate has increased between during this pandemic. Before that, it was something lower than during the pandemic. I hope.
Yes.
What else tends to do this?
I believe that more and more people are writing their paper and they are publishing their work at the moment, and I believe so. And two more important thing is that we have recently published two articles on. Those are actually basically one is a review article, and another is hypothesis for the COVID-19, that how stem cells could be used to overcome the COVID-19 pandemic. So I believe that those two are playing a good, major role. Those are getting good number of citation for us. So if you are aware that recently in China, 11 patients have been treated with the COVID with the mesenchymal stem cells, and they have shown a very good result. So that paper could be found at those were the paper was published in the journal Aging Cell, I believe. So there you can find it. So based on that, we further took up our hypothesis, and we think that if we will prime the stem cell, a stem cell priming is when you give some sort of challenges to the stem cell. For example, if you prime them with hypoxia, or if you prime them with some toxic chemical, then they have more potential to fight back So if we will transplant those primed MSCs to the COVID patient, they will be able to produce more cytokines, inflammatory kinds, and they will be able to more retain into the system. This is what we think, and I think that paper is getting too much of citation. So I think that would be a possible reason.
Yeah, fine, sir. Is your PhD is based on stem cell therapy?
No. So my
Yeah.
PhD was on the profilability effect of herbal treatment on stroke. But since my PhD, I am attached with the stroke recovery only. But eventually, I during my PhD, I got a couple of fellowship through which I moved to Germany and later on to Australia during my PhD. However, I have submitted my PhD in Jamia Hamdard. I started there in Jamia Hamdard, and then I finished up in Jamia Hamdard itself. So I chose two of the labs who were working on the stem cell during my PhD, and then I went there to be trained on stem cell therapy for ischemic stroke. And eventually, later on, I got both of my postdoc on stem cell therapy for stroke. And for the last 5.5 years, I'm working on as an independent faculty back in India on adult stem cell therapy for ischemic stroke. So
Yeah.
our another interest is in spinal cord injury. So we work on injury models, So as I have shown in one of our slide, that stroke, spinal cord injury, traumatic brain injury, they come into the injury model. They are not disease like Alzheimer and Parkinson's, Huntington, whatever it is due to the genetic makeup of the patient, and basically this is due to the neuronal degeneration, and these are diseased actually. So there is some mutation, and that ultimately produces leads to degeneration of neuron. But the spinal cord injury, the traumatic injury, and the stroke, it is not due to the any sort of mutation. It is due to the it is a sort of they are all three are comes into injury. So my lab works on the injurial aspect of the brain.
Yeah. I think this is how you jumped into the field of stem cell therapies.
Yeah, exactly.
Okay, so before you shining off, what is your last message for our aspirants belonging to different field of life sciences? What is your take-home message for them?
I will say that do very hard work, whichever subject you have opted for, because ultimately that is the only thing that is going to give you a recognition. I mean, there is no shortcut to hard work, So this field is very exciting, especially the the neuroscience. I'm not saying that you should opt for a neuroscience for post MSc, but neuroscience has a long future,
Yeah.
And finally,
Uh,
again, I would like to thank you, Partha, for this cause, the cause of education that you are providing to all of them. I am very delighted that I have seen today that approximately 3,100 people have subscribed your platform. So Gandhiji once said,
on all your future endeavors.
thank you very much. Thank you very much. And the
Uh,
last thing is that if anyone has any question, anything which is still there in their mind, and if they want to ask, please do write to me. And if you are coming to Lucknow, do visit our lab.
yeah. Thank you, sir. Thank you
Thank
so
you.
much, sir, for your time and patience in listening and answering all our questions. I hope all participants are satisfied with the speaker's elaborate explanation and answering all our questions. Thank you so much, participants.
Thank you
And
very much.
I thank you, today's speaker, Dr. Syed Raza, for spending your valuable time with us. And special thanks that you have made a video for us, animated video for us, and you have delivered to our team earlier, which made us more enthusiastic with those stuff, sir. Thank you so much, sir. You keep supporting us all in all our future endeavors. Thank you so much. I extend my sincere gratitude to you on behalf of my entire biomix team.
Thank you.
Thank you. Yeah, thank you once again. Thank you all for this. And I thank everyone who joined with us for today's talk. Thank you, everyone. Thank you. Thank you, so. Thank you. Thank you.